Another type of bacteria, there are the heterotrophic eubacteria. They do not make energy, they have to eat, unlike the cyanobacteria, unlike the autotrophic photosynthetic organisms. And when we talk about bacteria, this is what we're largely thinking of. These bacteria are decomposers. They break down organic material, they reintroduce it into they reintroduce it into the food cycle. Some are producers, such as azobacteria and rhizobium, and these are some examples of nitrogen-fixing bacteria. There's also E. coli and lactobacillus, which are typical human inhabitants. We have those in our body. E. coli is in the digestive tract, makes up a large portion of fecal matter. Lactobacillus is in the female genital tract and actually blocks out, by having its numbers there, other bacterial and fungal growth. Also, lactobacillus is important in breaking down dairy products, so it is used industrially in the manufacture of cheese, and sour cream, and yogurts, and things like that. A couple of other microorganisms, ones that are used in medical environmental applications. An example is the Pseudomonas. Another one is the Serratia marcescens. This is an opportunic pathogen. Actually, Alexander the Great's victory at Tyre can be credited in large part to this microorganism. It showed up in some bread, and someone read that as an indicator that there would be, they would win a bloody battle. And so it was a positive omen. As you can see, this does have these bright red colonies, and so it looks a little bit like blood. This also showed up on communion wafers, and it was thought of being the blood of Christ. Because it does have this bright red colony, this can be sprinkled into the atmosphere. You can have petri dishes setting out, and you can kind of see the flow of the air by where you get colonies of this microorganism. So this has been used to trace what would happen if a microorganism were dropped in certain air patterns, who would be affected. Another group of microorganism, bacteria, we have spiral and filamentous bacteria. The spiral bacteria include spirochetes and spirulia, and these are organisms that cause some human pathogens, specifically syphilis and Lyme disease. Now, the filamentous bacteria include the actinomycetes, which form strands of individually functioning cells. So even though they are in a strand, they are not tied to one another. Within the actinomycetes, streptomycetes, which are used to make a number of drugs, including tetracycline, erythromycin, streptomycin, neomycin, so on and so forth. Another group of bacteria are the gliding and sheath bacteria. Gliding bacteria actually produces slime, and their membranes undulate in such a way that they just kind of glide across this slime layer. It's almost like the movement of a snail. There are two important genera that live in some sulfur-rich muds, and these can break down these sulfur compounds and actually release them back into the sulfur cycle of the ecosystem. We also have myxobacteria, which can survive. It doesn't produce a spore, but it produces a spore-like body, which allows it to survive until conditions become better. We have sheath bacteria, and what happens with them is they have their cell walls, but it's also enclosed in an additional sheath that can protect the organism from predators. Among this is the genus Spherotylus. Just so you know, I will not ask you to name the microorganisms within these categories, I would want you to know what the categories are and the important characteristics of each of those categories. After that, when we talk about spiral and filamentous bacteria, I do feel that you should know that syphilis and Lyme disease are caused by spirochetes. Okay. There are also predatory and other classifications of bacteria. Delovibrio prey on other bacteria. They actually go in, work their way into the cell wall, populate that, kill the microorganism in the process. There's bacteroides, which is gram-negative, which lives in atmospheres that don't have any oxygen, but they assist in breaking down cellulose, and there are very few microorganisms that can do that. There are chemolithotrophic bacteria. These live off of, in this case, nitrogen, nitrobacter. They bring or put nitrogen back into the nitrogen cycle. We have acid-fast bacteria, and among these are the mycobacteria, which cause tuberculosis and leprosy. We will talk about these pathogens a little bit later on, but I would ask that you know that tuberculosis and leprosy are caused by mycobacteria. There are some bacteria that are so small we can't even see them with a light microscope, or can't see them well. These include rickettsias, and rickettsias are responsible for Rocky Mountain spotted fever, chlamydia, and one of these causes STD. And then there are mycoplasmas, which are so small they don't even have a cell wall. They have the phospholipid bilayer cell membrane like other cells, but they have no external structure beyond that. Work the way they do. Koch's postulates, all about associating a particular disease state with a particular microorganism. And so this was all first observed within that golden age of microbiology. So there are pathogens that are sources of disease. They cause their disease a number of ways. By growth. They can overwhelm a system in their growth, or overwhelm the system with a byproduct that they made, which can include toxins. So there are food poisonings that can occur with bacteria. By inducing a really overactive immune response, which can cause damage as well. So there are a lot of things that can occur pathogenically with these microorganisms. It's usually a byproduct of their growth in numbers. If we can stop their growth, their numbers, then the pathogenicity we can overcome. So, just to sum up what we've been talking about in this chapter, a few things to be aware of that we did not cover. You should read your textbook for clarification of any topics that you're not sure of. You can always email me and put questions on the questions and concerns part if you need additional clarification. Again, I cannot stress how important it is to learn terminology. You can't get anywhere if you don't know the terms. So learn the key terminology listed at the end of the chapter. Again, hope this will help you. Thank you very much.